Etienne Holder. She is a PhD candidate in biostats and epidemiology with mentorship from our own Dr. Sue Hankinson.
Good afternoon. I'll be talking today about estrogenic activity and postmenopausal invasive breast cancer risk. Estrogens are well known to increase breast cancer risk through ER-mediated pathway activation. Plasma levels of several endogenous estrogens, including estradiol, estrone, and estrone sulfate, here we refer to them as classical estrogens, have been consistently associated with an increased risk of postmenopausal breast cancer. However, it's important to note that other compounds also bind to the ER, such as environmental compounds. In our project, we were interested in examining whether a broader assessment of compounds activating the ER as a group would be more strongly associated with breast cancer risk. In order to do this, we evaluated the association between the results of a novel plasma bioassay that measures ER pathway activation with risk of invasive breast cancer. We used data from the Nurses' Health Study cohort, which began in 1976 and is ongoing. Women in our study donated blood between 1989 to 1990, and that was archived in freezers for later use. The cases and controls were selected during 8 years of follow-up after blood collection. We used a nested case control study design that included 371 cases and 731 matched controls. To be included in our study, women had to be postmenopausal, not using hormone therapy at blood draw, and have available classical estrogen measures. Breast cancer was confirmed via medical records. Estrogenic activity was assessed using a transfected human breast cancer cell line with a luciferase reporter. The animation here shows the mechanics of that bioassay. After treatment with our Nurses' Health Study plasma samples, estrogenic compounds in the plasma activate the estrogen receptor, which increase luciferase expression, resulting in a light signal that quantifies estrogenic activity. Our primary exposures are the fully measured bioassay, and then 2 derived bioassay measures representing estrogenic activity after removing the variation due to estradiol, the primary estrogen, and alternatively, after removing the variation due to all 3 classical estrogens. We use conditional logistic regression for our analyses, and these are our results. After accounting for confounders and other relevant breast cancer risk factors, we observed a near 2-fold increase of risk in comparing the high versus low quartiles of the bioassay, and this magnitude of association was similar to what we see for the 3 classical estrogens in breast cancer. The association was attenuated after removing the variation due to estradiol, and no association was observed after accounting for the variation due to all 3 classical estrogens. Now, when we assessed this association with ER-positive breast cancer, the results were essentially the same. So our findings suggest that other compounds contributing to ER activation cumulatively do not provide information on breast cancer risk beyond measuring classical estrogens alone. And while it may be true that there is no association, there are also possible reasons that we could have missed a real association, including issues related to measurement error. I'd like to thank our research team and our collaborators, and thank you everyone for listening.
All right, great job. I have a question to kick things off. Just wondering how correlated is the estrogen bioassay with the other specific estrogens, the E2 and the 3E? Are they highly correlated, and could that explain why controlling for those specific estrogens attenuated your initial findings?
So the bioassay, as you see from this chart here, is relatively correlated with estradiol, estrone, and estrone sulfate. And once we removed the variation due to estradiol as well as the classical estrogens, we see that the correlation is pretty much completely removed. So I'm not sure that that is the reason why we saw no association. Interestingly enough, once we looked at the bioassay with our 2 derived bioassay measures, we see that they're about 0.87 and 0.84 correlation. So there's still some variation going on that our analyses deemed not associated with risk.
Okay, thank you. We have a question from Laura Vandenberg. Can you talk about the choice of plasma rather than serum? which is often used to evaluate environmental chemicals with estrogenic activity?
I know that the Nurses' Health Study has plasma available for analyses. I'm not 100% confident in why we chose plasma over serum, but that's something I can definitely investigate and email you at a later time.
Okay. I guess another question for me while I'm waiting for other people to chime in. Based on your results, do you think this bioassay has any clinical relevance? Or is it better to use the specific estrogens?
I think it has similar, I mean, I think what we currently use now is just the specific estrogens is sufficient. I don't think the bioassay is adding anything additional. So I think what we currently use is sufficient.
Okay. We have another question, Sarah Memo. Can you translate this into practice for me? Are these estrogen measurements used clinically to screen for high-risk patients?
These estrogens, yeah, estrogen levels within postmenopausal women, they are used to screen for high-risk patients, as well as family history of breast cancer and other breast cancer risk factors.
Okay. All right, the dean just submitted a question. How is age and time of menopause controlled for? Excuse me, how is
So
age at time of menopause?
it was included as a factor that we adjusted for in our analysis, natural age at menopause.
And did follow up with that, did age of menopause influence, was it associated with the estrogens and the outcomes?
I have a table that shows some of our characteristics, and age at menopause, cases were slightly older at age of menopause. And while it is statistically significant, I don't think it influenced the results a great deal.
Okay. Any other questions out there? I'm not seeing any raised hands at this point. Okay. All right, I think we'll wrap it up there. Thank you very much, Etienne.
Thank you.
And we'll move on.